Welcome back to this next video, and in this video we are going to talk about a special class of proteins which are known as the Karyopherins. Now the first thing is, what are Karyopherins? Now these Karyopherins, they are a group of proteins which are involved in transporting molecules between the cytoplasm and the nucleus of a eukaryotic cell. When you talk about the eukaryotic cell, they have got a well-defined nucleus, and the material they should be transported between the cytoplasm and the nucleus. So you need a well-defined system for doing this particular function. And the Karyopherins are actually responsible for this transportation mechanism of the materials between the cytoplasm and the nucleus in the eukaryotic cell. Now, why they are called is the Karyopherins, the inside of the nucleus, that is called is the karyoplasm, and in this particular reference they are sometimes called is the Karyopherins. Now what these Karyopherins do, they generally mediate transport through the nuclear pores, which acts as a gateway into and out of the nucleus. And most proteins, they require these Karyopherins to traverse the nuclear pore. When you talk about these Karyopherins, as you are talking about the transportation between the cytoplasm and the nucleus, so these Karyopherins, they are actually divided into two broad classes. One that is known as the Importins, and these Importins are actually helping proteins to get into the nucleus. What I mean by this is that these Importins are responsible for transferring the molecules from the cytoplasm into the nucleus of the cell. And the second class of the Karyopherins, they are known as the Exportins, and the function of the Exportin, that is opposite to the Importin, that is that they are responsible for transferring a material from the nucleus into the cytoplasm of the cell. So when you talk about these Karyopherins, they actually belong to the Nuclear Pore Complex Family in the transporter classification database, and the energy for this particular transport, that is the movement of molecules from the cytoplasm into the nucleus or from the nucleus into the cytoplasm, that is actually derived from a Rain gradient that we will be discussing in detail. Now in this particular video, I want to focus only on the Importins. So the Importins, that is a type of protein, a class of the Karyopherins that move other protein molecules into the nucleus by binding a specific recognition sequence called the Nuclear Localization Signal. Now, if you talk about the proteins specifically, so in the cytoplasm there are a variety of the proteins, and not all of the proteins, they are required in the nucleus of the cell. Some specialized kind of the proteins, they are needed into the nucleus of the cell, but as the synthesis of the proteins that occur in the cytoplasm of the cell, so there should be mechanism for moving these synthesized proteins from the cytoplasm into the nucleus of the cell. And these Importins, they are actually moving these proteins from the cytoplasm into the nucleus of the cell. So how these Importins, they actually identify which proteins have to be moved from the cytoplasm into the nucleus and which protein doesn't have to move from the cytoplasm into the nucleus of the cell. Now the particular proteins which have to be moved from the cytoplasm into the nucleus of the cell, they have got a specific recognition sequence, and that particular recognition sequence is called is the Nuclear Localization Signal. Now if you focus on the name, so this actually say that this is a localization signal, and this localization signal is for the nucleus. So the nuclear localization signal is actually responsible for localizing a protein into the nucleus of the cell. So the Importins, they are actually recognizing these nuclear localization signal, and those particular proteins which have these nuclear localization signal, they will be moved from the cytoplasm into the nucleus of the cell. Now this nuclear localization signal, for short, which is known as the NLS, so this NLS signal, it consists of one or more short sequences of positively charged lysine or arginine, and these positively charged lysine or arginine, they are actually exposed on the protein surface. So if the NLS is present on the surface of the protein, it makes it very easy for these Importins to interact with them. Now this Importin, it have got two subunits. One is known as the Importin-α, and the other one is known as the Importin-β. So you actually have two subunits in the Importin family, one the Importin-α, the other one is the Importin-β. Now, the member of the Importin-β family, they can bind and transport cargo by themselves. What I mean by this is that the Importin-β, it doesn't need the help of any other importin to transport the protein from the cytoplasm into the nucleus of the cell, because this Importin-β, they can do it by themselves. The reason the Importin-β can do this, because they have got these heat repeats, and we will discuss in detail the function of the heat repeats. At this particular time, just keep in mind that the Importin-β, they can bind and transport cargo by themselves. But these Importin-β, they can also form heterodimers with the Importin-α. One Importin-α, one Importin-β, so you will be getting a dimer. As the Importin-α and the Importin-β are different from each other, we are using this term hetero over here. So one Importin-α and one Importin-β combines and it forms a heterodimer. Because the Importin-β, it forms heterodimer with the Importin-α, because the Importin-α cannot transfer the proteins from the cytoplasm into the nucleus by themselves. The reason the Importin-α cannot do this, because the Importin-α do not have these heat repeats. So when the Importin-α is involved in the transport of the cargo from the cytoplasm into the nucleus of the cell, they will be interacting with the Importin-β, because the Importin-α lack the heat repeats. Now, when this heterodimer forms, the heterodimer of the Importin-α and the Importin-β, so in this particular heterodimer, the Importin-β is responsible for mediating interaction with the nuclear pore complex, utilizing its heat repeats, while the Importin-α, it acts as an adapted protein to bind to the NLS on the cargo through the classical NLS import of proteins. So in this Importin-α beta heterodimer, the Importin-β is actually responsible for binding to the nuclear pore, and the Importin-α bind to the cargo protein, utilizing the NLS of that cargo protein. Now, if you focus on the structure of the Importin-α, we will discuss this later on, that the NLS Importin-α, Importin-β, when this trimer, it actually moves from the cytoplasm into the nucleus of the cell, there it dissociates after binding to the range GTP. We will discuss that in the later on in this particular video. Now, I want to focus on the structure of the Importin-α. If you focus on the structure of the Importin-α, on the N-terminal of the Importin-α, there is a particular region which is known as the Importin-β binding domain, and for short that is known as the IBB. So this yellow box I have made over here, this whole yellow box is actually the Importin-β binding domain, and this Importin-β binding domain is actually a 90 amino acid long. Within the Importin-β binding domain, there is a small subdomain which is known as the autoregulatory region, and this autoregulatory region is actually responsible for regulation of the activity of the Importin-α. So at the N-terminal there is an Importin-β binding domain, and within the Importin-β binding domain there is an autoregulatory region. So this yellow one, this big one, is the Importin-β binding domain, and within this Importin-β binding domain, this small red box is actually the autoregulatory regions. Now, the proteins that are transported from the cytoplasm into the nucleus of the cell, they can contain, they may be monopartite or they may be bipartite NLS motif. What I mean by that is there can be certain proteins which have one NLS, one nuclear localization signal. There may be protein which may have two nuclear localization signal. So if they have got one nuclear localization signals, they are actually known as the monopartite, and if they have got two NLS or two nuclear localization signals, they are known as the bipartite. Now the Importin-α, it actually contains several ARM repeats. These ARM repeats are actually the repetitive amino acid sequence of about 40 residues in length. So when you talk about the Excuse me. So when you talk about the Importin-α, there are several ARM repeats or the Armadillo repeats. Now these several ARM repeats, they actually form a curving structure, and this curving structure, it actually have got two NLS binding site within a shallow groove. And within this shallow groove or this curved structure, these two NLS binding site, one is known as the major NLS binding site and the other one is known as the minor NLS binding site. So what you can say is that because of the presence of several ARM repeats, there is a curving structure is formed, and this curving structure have got two NLS binding site. One is known as the major NLS binding site, the other one is known as the minor NLS binding site. Now, if you look at the location of the major and the minor NLS binding site in the Importin-α, their location is a little bit different from each other. The major NLS binding site, that is very close, that is very close to the N terminus of the Importin-α. If this is the N terminal, so here you can actually expect the major NLS binding site, which is close to the N terminal of the Importin-α. And it binds it can actually bind a monopeptide. What I mean by this is that the major NLS binding site, it can actually interact with a single nuclear localization signal, as well as one of the motif of the bipartite NLS cargo. As I've told you, there can be proteins which actually can have one NLS, or there can be protein which have got two NLS. So if a protein have one NLS, that is going to bind to the major NLS binding site. But if a protein have got two NLS, two nuclear localization signal, one is going to bind to the major NLS binding site, and the other one, which is near to the C terminal, that is going to bind to the minor NLS binding site. So the location of the major NLS binding site and the minor NLS binding site, that is different. The minor NLS is close to the C terminal, the major NLS binding site, that is actually close to the N terminal. Now this, if you talk about the Importin-β, now this Importin-β, this is a helicoid little molecule constructed from 19 heat repeats. Now what are these heat repeats? Now this heat repeat in the Importin-β, that is a special protein domain, and different kind of the protein that contain these heat repeats. They are the RAN GTP binding Importin and the Exportin cargo transport proteins. So these are just two of the examples where the heat repeats, they are present. The RAN GTP protein, they have got these heat repeats, and the Exportin protein that we'll discuss in the next video, they actually have got these 19 heat repeats. Now, if you talk about this heat repeat, it have got a signature motif by which you can actually recognize this heat repeat. And the signature motif of the heat repeats is the presence of conserved aspartic acid and arginine residues at position number 19 and 25. If you have got a domain and if you are saying that it is a heat repeat, so the signature is that there will be an aspartic acid at position number 19 and there will be an arginine at position number 25. Now what these heat repeats do, they are actually responsible for binding to the nuclear pores which are present in the nuclear membrane. Now many nuclear pore proteins, they contain a particular sequence which is known as the FG sequence, and this F is for the phenylalanine and this G is for the glycine. So the many nuclear pore proteins, they actually have this FG sequence of this phenylalanine or the glycine sequence. Now the heat repeats, they actually interact with these FG sequences. So this means that the Importin-β, it utilizes its heat repeat to interact with the nuclear pore proteins, and if it can interact with the nuclear pore proteins, it means it can actually move from the cytoplasm into the nucleus of the cell. As I've told you that the Importin-β, they can transfer the cargo from the cytoplasm into the nucleus by themselves. The reason is because they have got the heat repeats, they can interact with the FG sequences of the nuclear pore proteins, they can actually transfer the protein from the cytoplasm into the nucleus of the cell. Now as the Importin-α, they do not have this heat repeats. So if they do not have the heat repeats, it means they cannot interact with the FG sequences of the nuclear pore protein, and therefore they cannot transfer the protein from the cytoplasm into the nucleus of the cell alone. So they will be using the heat repeats of the Importin-β, thereby they make the complex with the Importin-α. Now, if you talk about the transfer, so if this is the cytoplasm, so what happens in the cytoplasm is that there is Importin-β, there is Importin-α, and there is the cargo, and this cargo, it can be your protein or it can be any other molecule, and this cargo, it actually have this NLS, this nuclear localization signal. So what happens in the cytoplasm is that the Importin-α, it utilizes its Importin binding domain to interact with the Importin-β, and it utilizes its NLS binding site to interact with the NLS of the cargo. So in the cytoplasm, you will be having this trimeric complex, in which there will be one Importin, there will be one Importin-α, there will be one Importin-β, and there will be your cargo protein. And the Importin-β in this particular complex will be utilizing its heat repeats to interact with the FG sequences of the nuclear pore to move this particular whole complex from the cytoplasm into the nucleus of the cell. Now, if you talk about the transportation of the cargo, this transportation needs to be unidirectional to be effective, because if you are interested in moving the proteins from the cytoplasm into the nucleus of the cell, and if this is a bidirectional process, it means that the protein can again move from the nucleus into the cytoplasm of the cell. So all of this exercise that will be a useless exercise then. So this transportation that needs to be unidirectional to be effective. Now this unidirectionality, it is actually achieved through the compartmentalization of a particular protein, which is known as the RAN protein. And this RAN protein, which is a small 25 kilodalton protein, that actually ensures that if a protein have to move from the cytoplasm into the nucleus of the cell, that will move in that particular direction. If a protein have to move from the nucleus into the cytoplasm of the cell, that will only move in that particular direction. Now, how this RAN achieve this unidirectionality? If you talk about this RAN protein, it can exist in two forms. It can exist in the RAN GTP form, and it exists in the RAN GTP form in the nucleus of the cell. The reason it exists in the RAN GTP form in the nucleus is because in the nucleus there is a particular protein which is known as the RAN guanine nucleotide exchange factor, or for short, which is known as the RAN GEF. So the RAN GEF is actually responsible for making this RAN into the RAN GTP form. Now inside the cytoplasm, if you talk about the cytoplasm, in the cytoplasm this RAN protein exists in the RAN GDP form, and it exists in the RAN GDP form in the cytoplasm because of the presence of another important enzyme which is known as the RAN GTPase activating protein, or for short that is called as the RAN GAP. So in the nucleus the RAN is in the RAN GTP because of the RAN GEF. In the cytoplasm the RAN is in the RAN GDP form, that is because of the presence of the RAN GAP in the cytoplasm. And now what happens is that these Importins, they actually release their cargo in the presence of the RAN GTP, and we will discuss that how it actually happens. So the Importin, they are actually releasing the cargo in the presence of the RAN GTP, and as the RAN GTP that is present in the nucleus, that means that the Importin will release their cargo in the nucleus, while the Exportins, they bind cargo in its presence. It means that inside the nucleus there is a RAN GTP. That RAN GTP will bind to the Exportin, and those particular Exportin will bind the cargo to move it out of the nucleus. Therefore, the predominance of the RAN GTP within the nucleus and RAN GDP within the cytoplasm, that actually ensures that Importin release their cargo within the nucleus upon interaction with the RAN GTP, and Exportin release their cargo in the cytoplasm upon the hydrolysis of the RAN GTP to RAN GDP. Now how this actually happens? As I've told you, in the cytoplasm, you are actually getting this complex of the importin-β, the importin-α, and the cargo, and the NLS of the cargo that actually interacts with the NLS-binding site of the importin-α, and the importin-β, it binds to the importin-β binding domain of the importin-α. Now, when this complex, utilizing the heat repeats of the importin-β, it moves into the nucleus of the cell, what happens is that inside the nucleus, the RAN-GTP that is going to interact with the importin-β, and when the RAN-GTP it interacts with the importin-β, this importin-β is actually dissociated from this trimeric complex. Now you are having the importin-β and the RAN-GTP binded to each other in the nucleus, and the importin-α and the NLS cargo attached to each other. So inside the nucleus, the RAN-GTP it interacts with the importin-β, and this complex of the importin-α, importin-β, and NLS cargo that is actually split into two. One, the importin-α is still bound to the cargo, and the importin-β that is now in interaction with the RAN-GTP. That means that we have released one extra component from the NLS cargo, that is the importin-β. Now we need the cargo in free form inside the nucleus. We do not want this in interaction with the importin-α. Now the next step we have to do is that we have to release this cargo from this importin-α. Now how this happens? Now in the nucleus, when the RAN-GTP it binds to the importin-β, this is actually causing a conformational change in the importin-β, and when there is a conformational change in the importin-β, that releases it from the importin-α bound cargo. Now, the N-terminal importin-β binding domain of the importin-α that contains an autoregulatory region that mimics the NLS motif. Now, come back to the diagram that we discussed at the start of the video, that there is an autoregulatory region within the importin-β binding domain. Now, when the importin-β that is in interaction with the importin-α, that means that that particular autoregulatory region that has been masked off, that has been blocked by the binding of the importin-β. Now, when the importin-β that is released by binding with the RAN-GTP, that means that that particular autoregulatory region that is now free. Now, as the structure of the autoregulatory region that mimics the NLS motif, so this auto-inhibitory region that it fold overs and compete for binding with the cargo at the major NLS binding site of the importin-α. So what happens is that when the importin-β that has been removed with the help of the RAN-GTP, the autoregulatory region that get exposed and that actually go and interact with the major NLS binding site, and when the autoregulatory region it interacts with the major NLS binding site, the cargo protein that is released from the importin-α. What happens next is then the cellular apoptosis susceptibility protein, which is actually an importin which is in the nucleus bound to the RAN-GTP that displaces the importin-α from the cargo. So if I get you back over here, and this particular site, initially when there is importin-β binding domain over here, that means that this autoregulatory region that will be blocked. When this particular autoregulatory region is blocked, that means it cannot perform its particular action. Now, when the importin-β domain that has been removed by the binding of the RAN-GTP, this means that this autoregulatory region that will now be exposed. Now, the structure of this autoregulatory region that is very much similar to the nuclear localization signal of the cargo protein. So, when the importin-β the importin-β that has been removed, this autoregulatory region it moves and it binds to the major NLS binding site, and when it binds over here, it actually displaces the cargo protein from the major NLS binding site of the importin-α, and with the help of another protein, the cellular apoptosis susceptibility protein, the importin-α that actually releases the cargo into the nucleus of the cell. Now, what happens is that when the the CAS and the RAN GTP that actually interacts with the Importin-α to release the cargo into the nucleus of the cell. This particular complex, the Importin-α and the CAS RAN GTP, it then translocate to the cytoplasm, and when it translocate to the cytoplasm, there is a particular protein which is known as the RAN binding protein. And what this RAN binding protein do, that it separate the RAN GTP from the Importin-α. So if you see over here that the cargo that is released by the interaction of two things: the interaction of the autoregulatory region with the major NLS-binding site, and the interaction of the cellular apoptosis susceptibility protein, the CAS, which is already in complex with the RAN GTP. So the complex of the CAS and the RAN GTP, this particular complex and the autoregulatory region, they go and they interact with the Importin-α to release the cargo. But in this particular case, the Importin-α that actually get in interaction with the CAS RAN GTP complex. So when this particular complex, the Importin-α and the CAS RAN GTP, it moves into the cytoplasm, there is another protein which is called as the RAN binding protein. This RAN binding protein, it actually interacts with this RAN GTP because this is a RAN binding protein, and when this interacts with the RAN GTP, it actually removes it from the Importin-α. Now this separation allows the access of the GAP, the GTPase activating protein. This GAP is then going to act on the RAN GTP, and when this act on the RAN GTP, that will actually be converted into the RAN GDP. Now, if this particular process continues, that if you are using the RAN GTP for the release of the cargo into the nucleus of the cell, and then that particular RAN GTP, it moves out into the cytoplasm because of this CAS RAN GTP, this means there will be a depletion of a RAN in the nucleus. Now, in action to prevent that depletion of the RAN from the nucleus, the RAN GDP that is formed into the cytoplasm because of the action of the GAP, that is transported back to the nucleus by its own import receptor, which is known as the NTF2. Now the RAN GTP that is going to interact with the NTF2, and this NTF2 is going to move the RAN GDP from the cytoplasm into the nucleus of the cell. Now what happens in the nucleus is, as I've told you, that there is this RAN GEF present, and what this RAN GEF will do, that it will convert the RAN GDP again into the RAN GTP, and this RAN GTP will then be responsible for releasing of the cargo from the Importin-α by the process that we just discussed. So this is actually the RAN cycle. What happens is that when you move the when there is the RAN GTP in the nucleus, and this RAN GTP moves into the cytoplasm by interacting with the CAS GTP, there will be the RAN binding protein, or this RAN BP, that is going to interact with the CAS Importin complex, releasing the RAN GTP, and then the RAN GAP is going to convert that into the RAN GDP. Now this RAN GDP will again move into the nucleus by the help of this NTF2 receptor, and inside the nucleus the RAN GEF is going to convert the RAN GDP into the RAN GTP for another round of the cycle. So this is all about the Importin, and we will discuss about the Exportin in the next video. So if you like the video, please subscribe to my channel, hit the like button, and share it with your friends.